So coming back to my presentation, I think in the morning, several of the speakers have emphasized the role of inflammation in cancer, specifically its role in inducing drug resistance, and a variety of targets have been identified. Just now also, one of the speakers was mentioning about CXCR4 in the morning, transglutaminase, or a number of other targets for it. So in my presentation, I will be talking mostly on one of the key enzymes of inflammation, that is cyclooxygenase 2, how it plays a role in promoting carcinogenesis and also specifically drug resistance. And as I was mentioning, inflammation is mediated by a variety of events. Among them, the lipid mediators are the most important. The prostaglandin E2 that is produced by the cyclooxygenase pathway, in which, so from the membrane phospholipids, by the action of the phospholipases, release of the fatty acid, that is arachidonic acid, takes place, which is acted upon by the inducible isoform of the cyclooxygenase, which is called as the COX2, also gets released, and that is the one which acts on the fatty acid substrate and produces prostaglandin H2. And this forms the intermediate metabolite, which is acted upon by various tissue-specific manner and produces a variety of mediators. The most important one is the prostaglandin E2, which mediates the inflammation, that is in terms of pain, fever, all other kind of symptoms. And today I will be talking mostly on this pathway. And as we know, non-steroidal anti-inflammatory drugs, they are targeting this cyclooxygenase pathway, and they are our favorite medicines. If you take only aspirin, we are consuming 120 billion aspirin tablets of 300 milligram size, and there are a number of other anti-inflammatory drugs. And even though they are there in the market, we know only for the past 40 years the mechanism of action, that they inhibit the prostaglandin biosynthesis and thus give you relief from all inflammatory symptoms. And the conventional anti-inflammatory drugs like aspirin, ibuprofen, all of them have the side effects, the most important being the gastric side effects. It was not clear how these side effects on one side and the relief from the inflammatory symptoms is taking place until 1990. It is in 1990 that we came to know that there are two isoforms of this enzyme. The COX1, which is constitutive, which is offering the protection from the protecting the GI tract through mucosal protection, whereas the COX2 is the one which is induced in response to any kind of stimuli, either injury or infection, and that is the one which acts on the arachidonic acid and produces the mediators which are responsible for inflammatory symptoms, whereas COX1 produces prostaglandins which are involved in housekeeping functions. And once this information was known in 1990, then using rational drug design approaches, because the crystal structure of both the enzymes was known by the time, and using this crystal structures by rational drug design approaches, COX2 specific inhibitors were developed as primary anti-inflammatory drugs, and they became the blockbuster drugs as soon as they were released into the market. And these COXIBs, a number of them came into the market. But very soon, see, these COXIBs are also not without side effects. So arthritis patients have to take these drugs throughout their life, and as on prolonged usage of these COXIBs, as you can see, there is increased incidence of cardiac attacks compared to the placebo. So that clearly indicated that COXIBs, though they are not having gastric side effects, but they have the cardiac side effects. So this story of anti-inflammatory drug discovery is going on. the effort is to come up with anti-inflammatory drugs which are not having gastric or cardiac side effects. And of course, this cartoon will tell you the story. If you go to the doctor with arthritis problem, he will put you on a COXIBs, and they may cause heart attack. A second prescription is to prevent the heart attack. And if you take that tablet, it may damage your liver. So the third is for protecting the liver. So this story of this kind of discovery of anti-inflammatory drugs without side effects is a major challenge. And now we came to know that when we are inhibiting the COX pathway, the substrate arachidonic acid is diverted towards the other pathway, that is lipoxygenase pathway, which is producing the leukotrienes. And this was shown to be responsible for the side effects, the cardiac side effects that were reported. And now the COX-LOX dual inhibitors appear to be promising anti-inflammatory drugs without gastric and cardiac side effects. And licofilone is one such COX-LOX dual inhibitor which is coming up into the market. So specifically with reference to the inflammation and cancer, so it was shown, the Time magazine has clearly indicated its importance, role of inflammation in promoting a variety of disorders, including the tumor. And if you see, many of the cancers, there is overexpression of COX-2. And as the progression, as the transformation from the normal epithelium to the carcinoma is taking place, neoplastic transformation, the COX-2 expression is also going up. And this overexpression COX-2 was shown to be responsible for a variety of the processes involved in promoting the cancer. And the COX-2 overexpression was shown in variety of cancers. And COX-2 inflammation has been shown as the hallmark of cancer. So inflammation plays a major role in cancer. In this talk, I would like to focus mostly on the how it is promoting the drug resistance, promoting the multi-drug resistance. And in this case, we have taken up chronic myeloid leukemia. As you know, that chronic myeloid leukemia is because of the chromosomal translocation, the famous Philadelphia chromosome, where part of the chromosome 9 is translocated to the chromosome 22, and as a result of there is a BCR-ABL kinase which got activated constitutively, and that was shown to be responsible for the chronic myeloid leukemia. And in fact, the first targeted drug, anti-cancer drug came into the market, that is the Gleevec, and this was shown, in fact, the most successful targeted drug for the cancers, and which paved the way for many other anti-cancer drugs targeting particular kinases. And where the Gleevec, that is imatinib, binds at the ATP binding site and thus blocks the activity of the kinase. And so in case of the chronic myeloid leukemia, and also in many other cancers, the resistance is because of a variety of mechanisms, either blocking the entry of the drug into the cancer cells, or pumping out the drug that enters into the cancer cells, or enhancing the metabolism of the cancer drug that enters into the body. These are variety of pathway, which in turn is involved in induction of COX2. And, um, and this was shown to be involved in mediating the overexpression of the multi-drug resistance protein and thus effluxing of the drugs that is taking place. This is one of them. So in a cartoon, as you can see, as the drug enters into the cell, it is being pumped out by the efflux pumps. So that was one of the mechanisms. And this, what we have shown in case of the cell lines. And then what we have done is we have collected the patients. So those who were exposed to, treated with the imatinib at different doses, 300, 400, 600, and 800 doses. And these are the control level of COX2 as well as MDR. As you can see, the expression of COX2 in all those who are undergoing chemotherapy is much higher. And the same is the case with MDR1, which is also higher. And in addition to the MDR1, there are other efflux pumps also, ABCC1 and ABCG2, which the other speaker was mentioning. They were also higher compared to the control in the various patients. So we collected about 40 patients. In all of them, there is at least majority of them, there is overexpression of ABCG2, ABCC1, MDR1 we have seen earlier. So in conclusion, the proposed mechanism is, I have not talked about the HDACs, histone deacetylases. Later on, we have shown that histone deacetylases through AP1 mechanism, it is involved in COX2 induction, which in turn produces the prostaglandin E2, and prostaglandin E2 is involved in overexpression of MDR1 efflux pumps. And this is the mechanism that was shown to be responsible. So this is the overall summary that when, in case of the efflux pumps is, if you are using COXIB, which blocks the COX pathway, then it also downregulates the expression of MDR1 and thus makes the imatinib-resistant cancer cells again sensitive to the drug. So this is the overall mechanism. So in conclusion, what we can say is the COX2 inhibitors can overcome the multi-drug resistance by downregulating MDR1 in case of the chronic myeloid leukemia. I have not presented the data here with reference to hepatocellular carcinoma. Doxorubicin-resistant hepatocellular carcinoma, also we have shown that the COX2 and MDR play an important role. Inflammation plays a major role. So use of anti-inflammatory drugs along with the standard chemotherapeutic drugs would be a good strategy to overcome the drug resistance. And we have a series of publications on these, not only on CML, but also on hepatocellular carcinoma. We have done not with Selective, and in addition, we also have developed a number of natural COX2 inhibitors, one being the C-phycocyanin, which also was shown to be involved. And this is our research team and the present team and our collaborators from different countries, the funding agencies, a lot of that from the industry. And I thank you all for your attention. Despite this being the last presentation, still I see large number of delegates. This shows the interest that people have on coming up with anti-cancer drugs or strategies for much better treatments for cancers. Thank you.